Welcome to AP Biofun with Dr. D, and we are on Unit 6, which is genetic expression. And today we are going to be talking specifically about the structure of DNA and how DNA gets replicated. The central dogma in molecular biology is concerning the flow of information in all living things, okay? And we know that DNA stores our genetic information, and that information is copied into a molecule of RNA via a process called transcription. The information in RNA is translated in a process called translation into proteins, and it is the proteins that do all the work in our cells: structure, regulatory, enzyme, signaling. So the information flows from DNA to RNA and from RNA to proteins, except in a few exceptions called reverse transcription, where information from RNA can be copied onto DNA. And we're going to be talking later in the unit about this. But today we're going to be talking about the process of copying the information contained in DNA for transmission to the next generation in a process called DNA replication. So let's examine the two both complementary and contradictory terms: genetic stability and genetic variability. So what does stability mean? Stability means we need to accurately transmit genetic information to the next generation with little to no change in that information. And genetic stability will ensure the short-term survival of a cell or a whole organism. And that depends on preventing changes in the DNA. The long-term survival of a species, however, requires that DNA be changeable over time. And these changes will provide genetic variation upon which selective pressures can act. And that's called genetic variability. It generates variety. No two offspring are the same. So how do we achieve genetic stability? Genetic stability is achieved by several different mechanisms: accurate duplication of the DNA during DNA replication. There is some proofreading and correction during DNA repair. And finally, accurate transmission of the DNA to daughter cells during cell division, which involves cell cycle regulation. And we talked about this in our Unit 4. How is genetic variability achieved? Now, genetic variability can result from the slow accumulation of genetic changes, which come from either uncorrected errors in DNA replication or actually a direct damage to our DNA, and these are called mutations. Genetic variability can result also from exchange of genetic material between chromosomes during sexual reproduction, which we call homologous recombination or crossover, and it happens in meiosis 1. We just learned this in Unit 5. Finally, genetic variability can also be introduced by mobile genetic elements, or so-called jumping genes, transposons, and viruses. And we're going to talk a little bit about this at the end of the unit when we talk about viruses. So why do we need DNA replication? Well, we know that already, right? Each time a cell divides, its entire genome must be copied and passed on to the new cell. You need to make 2 copies so that each cell has 1 copy of its entire genome. And genome simply is a term that refers to the collection of all genetic material in a cell. Our genome is comprised of 46 chromosomes or 46 DNA molecules, 23 from one parent and 23 from another parent. So how does it all work? Well, the double helix is really made from 4 different building blocks or monomers. They're called deoxyribonucleotide triphosphates, or DNTPs. Okay, so each unit is composed of a nitrogenous base, A, C, G, or T, a sugar, which in this case is deoxyribose, and 3 phosphate groups. 2 of the phosphate groups are removed. They're cleaved during synthesis. The bases are inside the helix and form hydrogen bonds with each other, which is the basis for complementarity, where A always bonds with T and C with G. The monomers themselves are linked via so-called phosphodiester bonds, and the bonds are formed between a hydroxyl group on one of the sugars and a phosphate group on the next. okay? So what enables DNA replication? And this is a very, very important concept, base pairing. So the complementarity of the bases really enables accurate DNA replication. One of the DNA strands can serve as a template for the synthesis of a new one. So whenever you have a G on the template strand, you're going to insert a C in the new strand. If you have a T, you're going to have an A. If you have an A, you're going to have a T. And if you have a C, you're going to have a G, and so on. The base pairing really it enables the accuracy of copying the genetic information. So in this model, what you will see here is that the original, the parental DNA double helix, is composed of two strands. We're going to call one strand S, and the other we're going to call S prime, the complementary strand. So if you were to unzip this double helix and use the S strand as a template to make a new strand, that new strand is going to be identical to the parental S prime. So you're going to be making a new S prime strand here in dark red. If you use the parental S prime strand as a template, you can now make a new S strand right here. So this enables accurate duplication of the genetic information. The fact that all living organisms, with the exception of some viruses, which are not considered living, use double helix DNA, double-stranded DNA, to safely store their genetic and transmit their genetic information underscores the significance of the structure of DNA, the importance of the structure of DNA in being in the ability to accurately copy genetic information. So DNA replication is semi-conservative, and the term semi-conservative comes from the two words semi, which means half, and conservative or conserve. Conserve means to keep. So it keeps half. Half of what? Each new DNA strand keeps half of the old one. So the parental double helix is unwound, and each new double helix contains one old and one new strand. So one of the original parental molecules and one new molecule. And you really saw this here. So the parental molecules are like in beige, and the new DNA molecules are one beige and one dark red, right? So it contains one old, one parental, and one new. That is called semi-conservative. Okay, now you might wonder how we know that. There have been some fascinating experiments done by two scientists called Meselson and Stahl, which demonstrated the semi-conservative nature of DNA replication. And feel free to read about this in your textbook. We're not going to be discussing this in this tutorial. Let's take a closer look at how DNA is made. Now, this is a polymerization reaction synthesis. So we're linking two nucleotides together to build a polynucleotide chain. And if we remember from Unit 1, sugars form rings. And these rings, we number the carbons, and we start with the oxygen around which the ring closes, and we go clockwise. So this is the first carbon, the second, the third, the fourth. And because this is a 5-carbon sugar, the 5th carbon is actually sticking out right here out of the ring. So this is 1, 2, 3, 4, and 5. Okay. So in this model, you see 2 nucleotides already linked, and a 3rd one is coming in. So let's look at this one right here. So on the 1st carbon of the sugar, that's where the base is attached. Here's a G, here's an A. On the 5th carbon of the sugar, we have a phosphate group. On the 3rd carbon of the sugar, we have a hydroxyl group. So the hydroxyl group of this nucleotide, the hydroxyl group, which is attached to the 3rd carbon in the sugar, is going to form a link with the phosphate group, and the phosphate group is attached to the 5th carbon of the sugar. So the sugar has a phosphate group attached to the 5th carbon. that phosphate group is going to form a bond with a hydroxyl group that is present on the 3' end of the sugar, on the 3rd carbon, right? So synthesis always occurs from, we say it occurs from 5' to 3'. Why? Because the last nucleotide in a growing chain has a 3' hydroxyl group. So the hydroxyl group is attached to the 3rd carbon of the sugar, and it's going to form a phosphodiester bond with an incoming nucleotide, specifically with the phosphate group that is attached to the 5th carbon, and so on. Due to the nature of this chemical reaction, we say that the growing DNA chain always grows from 5 to 3. So DNA chains have a directionality. There is the 5' end, which has a phosphate group exposed of the first nucleotide, and a 3' end, which has a hydroxyl group exposed of the very, very last nucleotide. So the double helix is directional, right? But the 2 strands, here's the catch, run in opposite directions. So one strand runs 5 to 3, the other one is flipped upside down, and now it runs 3 to 5. So whenever you have a 5 here, you're going to have a 3 here. So the structure is beautiful, right? It allows for faithful duplication, but it also presents certain difficulties. And these difficulties have been very elegantly resolved by a number of proteins we call collectively the replication machinery. And I would just like to remind you that evolution doesn't give you the best solution, doesn't give you what you need, it gives you what's available. So it's always a compromise. So while DNA structure has evolved to be perfectly suited for accurate duplication and transmission of information, it is also not perfect in terms of that it presents difficulties in how this is achieved. So let's look closely at, here we're looking at 2 base pairs, so 2 bases, 2 nucleotides on one strand and 2 nucleotides on the other strand, just so we can illustrate what it means to be anti-parallel, what it means that the 2 strands are running in opposite directions, okay? So in this ring here is the sugar ring, the oxygen in the middle, and again, we're gonna start, let me just annotate, we're gonna start counting. This is 1, 2, 3, 4, the 5th carbon is sticking out, okay? And there will be a phosphate group here. This is the 3' hydroxyl group, which forms a bond with a phosphate that is attached to the 5th carbon of the next nucleotide. All right. And there's going to be a 3' end here because this is the 3rd carbon here, the 3' carbon, which has a free hydroxyl group, right? The bases are attached to the 1st carbon. Okay, here we have a G and here we have an A, right? The opposite strand, what you can immediately see is that the sugar is flipped. Here the oxygen points upwards, here the oxygen points downwards. So everything is flipped. And what is sticking out here is the 3' end, the 3rd carbon and its hydroxyl group attached. So the strand is running in the opposite direction because everything is flipped upside down. Now you may wonder why that is. And without going into too many details, I will tell you that it has something to do with the way the bases match up, the G and the C and the A and the T. So the way they match up, they need to line up in a certain way and form the hydrogen bonds, dictates the fact that you need to flip the sugar upside down so that the C now matches up very well with the G on the opposite strand. Okay, so what are the problems presented by the structure of DNA? Well, the bases are on the inside of the helix, right? So we need to expose the bases because the bases are what is being read, what contains the information. So we need to unwind the double helix to expose the bases. We also need the 2 strands to keep them unwind. We need to keep them from reforming the double helix. And of course, we need to relieve tension, which is generated by the unwinding. And if you don't believe me, just take a elastic band, make a helix by rotating it, and then try to unwind it and see what happens behind the unwinding. Okay, so how do we unzip the double helix? Now, hydrogen bonds, we've learned about them in Unit 1. We know they're weak interactions. They're not really strong as covalent bonds. So you would think it would be easy. Problem is that DNA molecules are large, and there are millions of nucleotides and millions of hydrogen bonds in them. So it will be very, very hard. It will require lots of energy to unwind a whole DNA molecule. And as an example here, I'm going to give you that human chromosome 21 is the smallest of all human chromosomes, and it contains 48 million nucleotide pairs. So even if we assume they're all A and T, that is 2 hydrogen bonds per pair, that is at least 96 million hydrogen bonds that need to be broken in order to unwind chromosome 21. So how are we going to solve this problem? That requires way too much energy, and it's just impossible. Where's the cell going to get that much energy to unwind, to break 96 million hydrogen bonds? Well, the problem is solved by the simple means of, like, we're not going to unwind the whole molecule. We're just going to unwind small portions at a time. So little regions of DNA are going to be unwound at a time. And the places where DNA gets unwound are called replication origins, because this is where replication originates. So there's a bunch of proteins called initiator proteins that will bind to specific regions of DNA and unwind the DNA. And what they form is they form something we call a bubble, right? So you see how the DNA here is melted just in a small region. If you cover with your hand half the bubble, what you will see is a replication fork. So a bubble is comprised of 2 replication forks, a Y-shaped structure, and each replication fork moves in the opposite direction. One is going to go to the right, the other one is going to go to the left. Okay, so origins, replication forks, and bubbles. Again, a bubble is really comprised of 2 replication forks. And what I want you to notice here is, in this cartoon, you will see 3 different origins of replication. That is because DNA molecules are large, and we have multiple origins of replication. If you just have one origin of replication, you're going to wait forever for this DNA molecule to be replicated. So DNA replication will happen at the same time at multiple different places. It'll start multiple different places because there's multiple different DNA replication origins. For instance, in humans, we have around 10,000 different replication origins. They don't all start together though, and you can see here that this bubble is smaller than this one, and this one is smaller than this one. That's because this one started last, this one started next to last, and this one started first. But that's really not important. Replication has started at several different places. The forks are moving. This fork is moving this way. This one is moving this way. This one is moving to the right. This one is moving to the left. And when this one moving to the right meets up with this one, which is moving to the left, they're going to merge. They're going to fuse. So these bubbles eventually are going to meet up. They're going to fuse, and you're going to have yourself a completely duplicated DNA molecule. Okay. In the bottom, what you will see is half a bubble, right? A replication fork. So how do we melt DNA at a replication origin? Well, the enzymes involved are called DNA helicases, and it comes from the word helix, the double helix. So the DNA helicase is a collection of several different proteins, and it uses the energy of ATP hydrolysis to break hydrogen bonds in small regions of DNA. Now, as it unwinds small regions of DNA, it creates tensions. So here is a cartoon to show you how this works. It's a little bit simplified, but you will get the point. As you unwind a portion of the DNA, On both sides of the unwinding, you would get supercoiling. It will create tension. And that tension needs to be relieved. And it is relieved by another set of enzymes called Topo Isomerases. It comes from topology. So Topo Isomerases are responsible for creating little nicks, little breaks in the DNA to help unwind the strand that has gotten like, it's like almost like getting like things into a knot, into a tangle. So these tangles are not good. This is tension, this is supercoiling, this is tangling. So it gets all messed up, right? So it needs to be untangled. And it's untangled simply by these enzymes, which are gonna just create a little cut. Oops, I'm sorry. Create a little cut to get rid of the knots and the tangles, and then re-glue the strands again. Reseal them, basically. Okay. Another important factor in the unwinding of DNA is single-stranded binding proteins. These are proteins that would cover the unwound strand to make sure that that strand doesn't seal back on itself or on the other strand. So DNA molecules, not just DNA, but RNA molecules as well, nucleic acids tend to spontaneously form double-stranded regions. What you will see in this case is this single-stranded DNA, even though it's not glued back up to its partner, it kind of folds on itself. And they're called like hairpins. I don't know why they're called hairpins, because they look like a hairpin. But regions that are complementary to each other will fold on themselves and form temporary double-stranded regions. And why is that? It's just energetically favorable for bases to pair. So this occurs spontaneously. And we need to prevent this because this is not a single strand of DNA. I mean, yes, it's one strand of DNA, but it has these like matched up regions. And this will prevent the enzymes that are supposed to be reading the bases and adding new bases to do that. So in order to keep that from happening, this unwound strand will be covered in these single-stranded binding proteins, which will prevent this from happening. Okay, so in summary, how did we solve problem number 1? Well, initiate proteins in DNA helicases will melt the helix at origins of replication. DNA Topo isomerase untangles, it relieves the tension. And single-stranded proteins will bind to the exposed single strand and keep it from reforming a double helix. Okay. Second problem in DNA replication. DNA polymerase, which is the enzyme that links the nucleotides, that actually makes the new DNA strand, is perfectly imperfect. Okay, that's the enzyme that catalyzes the polymerization reaction. It is very efficient and it works very fast. And it's also very accurate. So what else could you want, right? Efficiency, speed, accuracy. Well, it turns out this enzyme is incapable of starting from scratch. It needs a paired nucleotide with a free 3' hydroxyl group to start synthesis, so it cannot start from scratch. So for this reason, DNA replication utilizes an enzyme called DNA primase. Sometimes you'll also hear it referred as to RNA primase because it really is an RNA polymerase. It actually will synthesize a short stretch of RNA to provide a free 3' hydroxyl end so that DNA polymerase can start synthesis of new DNA strand. And you will see here in this model, the primers in green, all right? These primers will eventually be erased, they'll be deleted and replaced with DNA. But they serve to prime, to get the DNA polymerase going. DNA polymerase cannot get going unless it already has some paired nucleotides ready. So DNA primase generates these little green things here, these little RNA primers, which eventually will be erased. All right, now let's look again at the anti-parallel nature of the DNA strands. I would like to remind you that one strand runs phosphate to hydroxyl, right? 5 to 3. Oxygens are pointing upwards. The other strand, the oxygens and the sugar are pointing downwards. See, it's flipped. So it runs 3' hydroxyl to phosphate, 5'. All right. And again, this is because of the way the bases have to pair. The 2 hydrogen bonds between A and T and the 3 hydrogen bonds between C and G. And the way the bases interact inside the helix is such that the 2 strands need to be flipped. Well, that's great, but I just told you that synthesis always occurs in the 5' to 3' direction, which means synthesis on one strand will occur this way, but on the other strand, it has to occur in the opposite direction. So how does that work, right? So this is problem number 3. The synthesis of a new strand of DNA is directional. It occurs in a 3' to 5', in a 5' to 3' direction. So how do we synthesize both strands at the same time if they run in opposite directions? Okay, how do we do that? That's a problem. Well, that problem is solved the following way. One DNA strand is synthesized discontinuously. It is called the lagging strand. There is a leading and there is a lagging strand. Okay, so let's look at half a fork. So the template strand here is 5 to 3. The primer is right here in green, and DNA polymerase is now going to synthesize from 5 to 3. It's going to synthesize in this direction. And this is the direction in which the fork is moving. The fork is going to be moving in this direction, and DNA polymerase is moving with the fork in this direction for this strand. But how about this strand? This strand is opposite. It runs 3 to 5. So synthesis has to go in the direction opposite of the way the replication fork moves. How do we do that? Now, if we were in a classroom, we would be demonstrating this. A dance. We've made up a dance called the dancing polymerase, but we're not. So now you're going to have to really, really imagine hard. So the 2 polymerase start right here. One of the polymerases is facing this way, and it's moving this way. The other polymerase is facing in the opposite direction. Why? Because it synthesizes that way, but it needs to move this way. So what is the polymerase going to do? The polymerase is going to jump back to here and synthesize this way. Then it's going to jump back here, synthesize this way, and keep jumping backwards, moving forwards. Or actually, it's going to be jumping forwards towards the direction of fork movement, but synthesizing backwards. So what the polymerase does is it jumps back, synthesizes, jumps back, synthesizes, jumps back, synthesizes. So it'll synthesize this strand in small portions, discontinuously, not in one continuous thing, but in little parts. All right. So the other fork, which is going in the opposite direction, will have the same problem, except because this other fork is moving this way, this one now it's going to be the leading strand, which is made continuously, and this one is going to be made discontinuously. These fragments, these fragmented syntheses, are called Okazaki fragments because they were discovered by a scientist named Okazaki. Okay, so we need to distinguish between leading versus lagging strand. Both new DNA strands are made in the 5 to 3' direction. The lagging strand is made by backstitching. Backstitching means the polymerase jumps backwards in the direction, backwards relative to the direction in which it is synthesizing, but really in the same direction as which the fork is moving. So the lagging strand is made in short segments, Okazaki fragments, and they're later stitched together to accommodate the direction of fork movement. On the lagging strand, the DNA primase also jumps backwards. in relation to synthesis, but forwards in relation to fork movement. And again, both DNA polymerase and primase are literally jumping backwards in relation to direction of synthesis, but really it is forwards when you consider fork movement. Okay, the RNA primase will be erased. There is an RNase H enzyme that will actually erase the RNase because it recognizes them as something foreign, something not DNA. And now DNA polymerase is going to fill the gaps. So wherever you had RNA primers, now DNA polymerase is going to be filling the gaps. All right. Once the gaps are filled, though, you will have two fragments which are not linked by a phosphodiester bond. So I want you to look at the very left here. This is one fragment and this is another fragment, and they're right next to each other, but they're not linked to each other. So you have a free 5' phosphate end and a free 3' hydroxyl end. And these two fragments need to be stitched together. They need to be stapled together. And they're stapled together with the help of DNA ligase. DNA ligase staples two adjacent DNA fragments using the energy of ATP hydrolysis. Okay, so DNA ligase is really necessary to stitch together both the Okazaki fragments on the lagging strand and different fragments generated by multiple forks. So DNA ligase functions on both leading and lagging strand. Okay. So let's summarize the replication machinery. What is needed to duplicate a molecule of DNA? Many different proteins cooperate, act in concert. Collectively, they form the replication machinery. We have the helicase, we have the Topo Isomerase, primase, polymerase, and ligase. We also have initiator proteins, single-strand DNA binding proteins, and a nuclease, an RNase that will actually erase the RNA primers. And of course, we need DNA template, and we need DATP, DCTP, DTTP, and DGTP, the deoxyribonucleotides, which are the monomers, the building blocks of the new DNA strand. Okay, so this picture basically summarizes everything that is happening in DNA replication. I am strongly suggesting that you click on this link on your own and watch a recording, a narrated animation of DNA replication to help you remember and understand all the details about DNA replication. And we're going to stop here and continue next time. Thank you.